Hello, welcome to lecture 8, that is metabolic engineering strategies in plants. So, in the previous class we talked about the approaches of metabolic phytochemistry for studying metabolic pathways. There I have discussed in detail how enzymatic route can be elucidated at biochemical level. I have also talked about the use of transcription factors to see the consequence on the specialized metabolites accumulation, and now we will try to design or categorize the different strategies through which metabolic engineering can be applied, aiming to produce the desired product at higher level. Here I would like to tell that when in the subsequent classes, when we will start the particular metabolic pathways. So, there we will study the pathways and then I will cite examples how to modulate, modify or suppress the pathways. So, there all the steps will be discussed in detail, but in this class I will try to give a brief overview of different approaches of metabolic engineering. So, here any particular case studies will not go into that details. Telling this, I just now go to the next slide. So, the concept covered how to make more of a desired product, how to reduce the level of unwanted compounds and how to make new compounds through metabolic engineering. So, the ultimate target of metabolic engineering is basically to produce the designer plants, which are capable to produce the compounds of interest using both using a choice of green and sustainable way. So, let me show you the different approaches for meeting the objectives of metabolic engineering. The first one is basically the more of a desired compound, how to make more of the desired compounds. So, under this what you can do, you have to increase the flask of the desired products. So, increase the flask means that the flow will move more in that direction, that ultimately leads to more product formation. So, that can be done by over expressing the rate limiting steps. So, if it is a short pathway, then that will be very easy, and when we will talk about the alkaloid metabolic engineering, I will give you a very successful example of such. Other one is that to reduce the flux flow through competitive pathways, and the another approach will be to over express the regulatory genes, such as I have mentioned about the transcription factors, and as a result of that you may get the desired products accumulated in higher amount. The other way to increase more of the desired compound is you can block or shut down the catabolism products. So, that means the inhibition of catabolism products, or another way to increase the number of producing cells, that may be another way to make more of the desired compounds. Now, new compounds can be produced in the plant by completely hiring a metabolic pathway which is not occurring in your target plants, and if you put that, that may produce the new compound, or you can you try to make a target compound enhanced by putting a desired gene, but what will happen as a result of that gene expression, that your target compound may get unaffected or even suppressed, but you will find another compound get enhanced, and that is because that may be a new compound. And why that happened? That is because there may be several branching steps which we are not aware of, and as a result of targeting a particular step may leads to upliftment of another step. So, sometimes through this way a new compound or novel compound can be harnessed. The third one is basically to reduce the level of compounds. So, where you can reduce the flux in a biosynthetic pathway, that means how you can do that, you can basically block the particular metabolic steps by antisense suppression or even RNA interference based approaches. The other way is that you can increase the flux of the competitive pathways. So, when you increase the flux of the competitive pathways, then what will happen? The product which you want not to produce in higher amount will definitely will get suppressed, because competitive pathway will take care of that and they will produce other products. Or you can also suppress the regulatory steps, that is, if a transcription factor is playing important role in upregulating the pathway. So, you can basically suppress the transcription factor genes, and as a result of that your desired product or your product level will be reduced. And also another way is that you can increase the catabolism. Now, here we will show the potential biotechnological exploitation of genes of plant specialized metabolism. Say a pathway, normal pathway is operating, which makes A to B, B to C, C to D and D to E, and also D can make F. Now, if you are interested to produce the one of the intermediate C in higher amount, then what you have to do? You have to block the downstream pathway, that is C to D onwards. So, that can be blocked. So, what is showing in the extreme right side by antisense suppression. Other way, if you want to make more of E, so the competitive route from D to F must be blocked. So, what is showing here, and if that can be done, then you can accumulate more of E. Now, it is also possible to utilize this endogenous plant pathway and transfer a heterologous gene, which is non-existent in the plant, and as a result of its expression it can produce a new compound, provided that the substrate of that enzyme, heterologous enzyme, must be available in the plant. In this case, the heterologous gene is expressed, which utilizes the substrate B and produces Z. So, here the B substrate will be competed by both the endogenous enzyme, which converts B to C, as well as the heterologous enzyme, which converts B to Z, and if you use a strong promoter, so that there is every possibility that Z will be accumulated in higher amounts. Now, what about this pathway expressing in microorganism? If it is a short pathway, then it is easier to produce this product, easier to transfer this pathway in the microorganism. So, like here it is showing that G1, G2, G3, G4, and of course, the plant genes can be transferred into the microorganism with the use of appropriate promoter, and the genes will be expressed, but that will not produce the product. The reason is why? Because microorganism will not get the substrate to channelize or to run the plant pathway like A to make E, because A is a very much plant substrate. So, what one need to do is basically grow that microorganism expressing these plant genes, and then you feed with the substrate, and as a result of substrate feeding, that is A. So, that will be subsequently utilized by the new pathways, and it will make more of E, that is possible. And also it is possible to do a single step reaction. For example, if you are interested only the producing D in higher amount, so the genes which convert or the enzyme which converts C to D can be expressed in the microorganism, and if the C is cheaper, that can be fed, and then as a result of that D is produced in higher amount. So, this is the normal plant pathway which produces product A1 and product 2, and if you want to, if you can block the side chain, then this product will produce in higher amount, and if you introduce a transgene, so then you see that is a blue one that produces a new product, and at the same time you need to block the other competitive pathway, and finally you can decrease the catabolism of the desired product like blocking the side path, and then as a result of that the product 3 will the product 2 will accumulate, and then you introduce a transgene, then it will make a new product 4. So, that means the new product 4 is indeed a new product. So, which was not produced by the plant, but when you introduce the transgene and block the side pathway genes, you can get the whole product. So, these are the ways the medicinal plants can be tailored to produce pharmaceutically important alkaloids, and in the subsequent classes we will see some successful applications and the limitations of such work. So, to brief up the things, that once a biochemical pathway is identified, it can be altered. How it can be altered? You can either block the pathway flow for the formation one particular pathway. As a result of that, what will happen, that when this will accumulate in higher amount, then it is possible that this pathway will move in this direction, that is showing in the pink arrow, and it produce E in higher amount, and when E is producing in higher amount, it is also possible that G and H can be produced, or you may have to introduce particular enzymes here, which can make more G and more H from E. So, gene function can be added or augmented environmentally or in the transgenic manipulations. So, this is all possible. So, in other words, a metabolic pathway can be transferred into bacteria, into yeast, as well as into plants and plant cell culture as well, and provided you have the right condition. So, you will be able to harness the desired product of interest. Now, there are few pros and cons, like introducing a pathway in plants, of course, very challenging, very demanding, but what happens that you never know, plant cells are very complex, there is a lot of side branching. So, if things work properly, then you may get the desired product P3 in higher amount, but also there is a possibility that the C, which will produce P, that the C may prefer to run the pathway towards D. So, it is also important that you may have to block here, or if you are also interested to produce more of E, then the catabolism of E to be blocked, what is showing here. However, it is easier to express the genes in the microorganism, but often what you will find that the substrate is not available. So, the feeding of the substrate is required, and this is only feasible if the cost of the substrate is low, so that the process can be scaled up and go to the industry. So, this I have already discussed. So, I am not going to discuss this in details. So, this is just another way of presenting this. So, I move to the next slide. So, this is a benchmark procedure to produce plant natural products in microbial cell factories. That means, the plant natural products are now produced in microorganisms by utilizing different strategies, and in several cases scientists were successful, and few products are almost now producing in the commercial scale. Here is that many medicinal metabolites have been the object of pathway engineering. So, for example, in bacteria and yeast. So, bacteria and yeast. So, these products are basically can be produced in the bacterial system by appropriate expression of the target genes. Similarly, some of the products can also be produced in the plants as well by appropriate targeting genes. So, this is basically just to show the range of compounds which can be produced either in bacteria or in plants or in the microorganisms. Okay. So, both plants as well as plant cell cultures can be targeted for production of the metabolites. Like plant cell cultures, the genes can be transferred into the plant cell cultures, and as a result of that, the plant cell cultures will be capable of producing your desired product in higher amounts, which is not the case in the normal wild type cells. Similarly, the pathway can also be transferred into the plants. For example, in lavender, which is producing essential oil, and upon expression of the desired gene, say this TXS, which catalyzes the early steps of the metabolic pathway of terpenes. So, what will happen as a result of that? You will get the altered composition of the essential oil harnessed from the transgenic plant, and this has happened because of the appropriate function of the DXS, which will basically flow more the metabolites into the pathway, leading to the formation of a range of monoterpenes. Now, this is basically a brief reviewing of the different process or different steps which can be produced using plant enzymes, using even in the human enzyme or even using the microbial enzymes. So, native pathway for the production of reticulin, which is showing in the black arrow, and the product of reticulin is showing in the red arrow. And for the conversion of reticulin to salutaridine, human P450 enzyme is used, so which is showing in the purple arrow. That means, a human P450 genes can be expressed into the plant, and as a result of that reticulin can be made into salutaridine, and which ultimately can be used to produce the morphine. An interesting point is this, that when when you use this metabolic engineering approaches, the reticulin in this case can produce both as both the racemic mixture. So, that is why both S and R are showing here. So, that is the advantage. So, it has more relaxed substrate specificity, so that it can be properly utilized by the subsequent enzymes and produce the product of interest. So, that means the metabolic engineering can be used for production of specialized metabolites in yeast, where you can hire the enzymes from different sources. So, this is another straightforward example, which the paper which published in Nature. So, as I have mentioned in the previous class about the caffeine. So, it is always desirable to reduce the caffeine content in coffee. So, what scientists did basically, they used the RNA interference approach, through which basically they have silenced the genes for caffeine biosynthesis. The first one is the theobromine synthase, which produce theobromine from 7-methylxanthine, and the other one is the caffeine synthase. So, what they did actually, they have blocked the theobromine synthase here, so that less theobromine is produced, and as a result of less theobromine is produced. So, the pathway for caffeine also get reduced substrate. So, eventually it produces less caffeine, and this is the wild type plant and this is the transgenic plant, and what is showing here is basically the less caffeine here in the transgenic line than that of the control. So, this is the caffeine level of the control lines. This is the theobromine and this is the caffeine level, right. So, these are the control and this is the transgenic. So, both the theobromine and the caffeine are getting suppressed, which is very much desirable in the coffee plant. Now, we will talk about the new strategies of plant metabolic engineering research. So, what you can do here? You can target a medicinal plant. So, where active ingredients need to be identified, and what you wanted to do, that should be very clear. So, the structural genes should be known and the key regulators to be identified, then the medicinal plants can be modulated for produce the desired product in higher amounts. Alternatively, if you can use the model plant to produce the desirable product, then what is required, that you have to hire the specific gene and then you have to use a specific or new promoter, so that it express well in the model plant, and there may be need for controlling the flux by either specifically overexpress a transcription factor there, or in some cases gene editing will be required. So, by combination of these two aspects, the new generation of metabolic engineering can be carried out through the construction of new biosynthetic pathways and use of transcription factors, and driven by specific promoters, manipulation of the flask, and even if required the deletion of the competitive pathway, competing branching pathways, and as a result of that, modern plants can be re-engineered for successful production of high value metabolites. So, in other words, that the genes from the medicinal plants can be hired, and with the appropriate experimental design, the model plants can be exploited to produce the desired product at higher level, which can be scaled up for subsequent hardness at the industrial scale. So, this is nothing but again what I have said in a nutshell, that what are the different ways to enhance the specialized metabolites. One is the endogenous enzyme overexpression, is already the pathway is there. If you overexpress, in several cases it worked well, but in also there are cases where sometimes overexpression of endogenous expression leads to gene silencing, which earlier people explained in the terms of co-suppression. Now, what is basically happening there is that RNA interference made blockage of the pathway, but in several cases these strategies were successful. Other ways that foreign enzymes can be overexpressed. So, if you take a heterologous gene, there are chances of functioning is very high, or the transcription factors can be overexpressed, or the gene can be silenced by RNA interference approach. The genome can be edited, and the edited genome may work well, leading to the formation of the desired product, or sometimes you can express multiple genes, and as a result of that you can get a range of desired products in higher amounts. Now, let us make an overview of different level of metabolic engineering. Say introduction of the new structural gene will allow production of specific metabolites. So, this is the structural gene which is showing in the yellow. So, if you introduce here, so this will produce the new gene. So, this is the level 1. Further addition of introducing transcription factor will upregulate the structural gene to synthesize more metabolites. So, that means if you induce the transcription factors, then what will happen? Then that may upregulate the upstream enzymes of the pathway, and eventually it will make more of the product. And the other one is that if you increase the supply of the substrate or energy or reducing power, that will enhance the downstream synthetic product. So, that means the at the precursor level, if you can manipulate, so that may make effect towards the end product, or if you supply a cheap substrate at the precursor level, then that may be taken up, and which eventually make the product in higher amount. And then what has been suggested that any specific promoters can be used, which can perform the job better than the existing promoter being used, or one can also delete the branching pathway by suppressing certain genes, and as a result of that you can also get the desired product in higher amount. So, combining of these 3 engineering levels will result in high level of accumulation of specific metabolites in higher directed manner. So, this was what is basically suggested by this group and the report published in Molecular Plants in 2018. So, now let me explain this multi-level engineering. Ultimate aim is to produce the isoflavonol genistein. So, this is basically a dietary phenolics. It is produced in the legume, and your aim is basically this to produce in the higher amount. The first one is basically the first level will be introduce the pathway gene. So, isoflavone synthase here, so which will make the genistein, but that may not work at the desired level. So, upon expression of this, the product you got enhanced, you are not happy with that. So, next what you should plan is basically you try to do the second level of targeting, where you will take a transcription factor gene that you can hide from a model plant. Here is Arabidopsis thaliana 12. So, that what it does basically, it will basically the upregulate the expression of the upstream genes of the flavonoid pathway like PAL, CHS, CHI. So, this we will learn details in the subsequent classes. Not only that, it will also upregulate the another pathway which makes the flavonol. Okay. And these transcription factors will also able to upregulate the primary metabolism, that means that the glycolysis when glycolysis converted beyond glycolysis, the product is converted to phosphoenol pyruvate the pentose pathway, and also this level where erythrose phosphate is converted into secumate, the first enzyme DAHP synthase. So, these two enzymes enolase and DAHP synthase, these are also being upregulated by MIB transcription factor. That means, this second level of upregulation by expressing transcription factor can indeed modulate both at the junction of the primary and specialized metabolism and also the upstream specialized metabolism. So, all this, and so this is now divided as a third level in red. So, that what I mean to say that this can itself modulate this, or if possible such genes can also be expressed, and as a result of that what you get, the desired product is accumulated at very higher level. So, this is what is called multiple level engineering for genistein, a dietary phenolics which is produced in the legume plants. So, there are several successful metabolic engineering strategies which can alter the content and composition of phytochemicals. For example, there are successful examples in terpenes, there are successful examples in phenolics, there are successful examples in carotenoids, there are successful examples in flavonoids, there are successful examples in alkaloids, also there are successful examples in the oils. So, few of these examples we will discuss when we will talk the metabolic pathways. Of course, we will not discuss all, but whatever we will discuss in detail, so that the concept will be clearer to you in through your attendance in the subsequent classes. So, now before ending, I will tell you the timeline of the production of span specialized metabolites in heterologous organisms. That means, once the gene was isolated or then the gene was expressed, and as a result of that the heterologous organisms, it could be plant, different plants, it could be microorganisms, they are capable of producing these new products which are basically not meant for producing in that particular targeted organism. And that work started from 2000 and until 2020, there are a lot of achievements. So, the report, the paper published in Natural Product Reports basically highlighted these achievements, this historical summary, and that is indeed very exciting. This is very exciting time. And not only that, there is a tale of two recent stories leading to widening and improvement of metabolic engineering for plant natural products. So, here the colchicine, colchicine is an alkaloid. So, that is produced in the flame lily. So, this colchicine now can be produced in the tobacco, and this colchicine complex pathway has been elucidated, and this paper published in the famous journal Nature. So, synthesis of a colchicine precursor in tobacco. So, that is a great achievement. And another one is that the tropane alkaloids, scopolamine and hyoscyamin, these are commercially produced and has have lot of pharmaceutical applications. This can now successfully produced in yeast, so that you need not have to depend on the plants. So, yeast can be grown in the bioreactor and these alkaloids can be harnessed, and this was also published in the work in Nature Communications in 2019. And the industrial scale production of benzyl isoquinoline alkaloid reticulin, which is basically the precursor of morphine. So, the industrial scale production has just started. So, using the yeast system, the pathway has been expressed in yeast, and this work has been reported also in this Nature. Before I end, there is one point to discuss here, that we are targeting either single enzyme or multiple enzyme, but one should know that these sequential enzymatic steps are sometimes organized in aggregate, and this organization sometimes they mostly retain or they stay in the endoplasmic reticulum and form the multi enzyme complex. Okay. So, if you try to target one gene, so what will happen? That may produce the desired product, but that may not really make modulation of the entire metabolome or that metabolic channel. So, the point is that the concept of single enzyme unlikely affect the flask of the entire pathway leading to the formation of the desired product. So, this was anticipated in early 90s, and in fact, this concept is very true, and that is why now there has been attempt to express multiple genes in the plants, but the problem is this, still the technology is a great challenge. So, transforming so many genes in the plants, particularly to integrate into the plant nuclear genome is difficult. In that case, maybe the plastid, transforming genes in the plastid will come as an alternative to solve this problem, and we will address this issue towards the end of this course, where we will see that how genes can be transferred into the plastid, where the entire operon can be transferred, and as a result of that the desired product can be produced. But eventually, but plastid cannot do all. So, eventually genes to be transferred into the nucleus. So, what we need to know? We need to know more of the basic science of this multiple enzyme regulation and their how the aggregate enzyme function, and once this is clear, more defined strategies can be designed to get the desired product in your in the level what you wanted. So, to conclude, although still we know very little about plant specialized metabolism and its regulation, this limited knowledge has been used quite extensively to explore the possibilities of metabolic engineering. Why I said so? Because I have mentioned that phytochemists they isolated several compounds, but the metabolic steps we have very limited knowledge. Unless we have a thorough knowledge, we cannot do metabolic engineering applications, and because of the more or less empirical approach, some unexpected result has been found. And also it is as I said that it is sometimes difficult to predict the result of overexpression of a single gene, because as a result of overexpression that may activate the other competitive pathways, and that may leads to sometimes undesirable product formation, which really is a nuisance sometimes. So, that we need to know more about the regulation, and regulatory genes for upregulation of competitive pathway may seems to be a promising approach, and work is also directed in this direction, and several positive outputs are coming. One example I have said in the previous class about the overexpression of the transcription factor leads to anthocyanin accumulation in tomatoes. So, such examples are very exciting. So, with this I end this class on the metabolic engineering approaches and the strategies which are used for manipulating the plant pathways, and in the next class we will talk about the how to transfer genes into the plants. So, the isolation of gene is beyond the scope of this class. So, as a metabolic engineer you will have the gene in your hand. So, that will be given by molecular biologist, and now these genes to be transferred into desired plant. So, the technology to transfer needs to be discussed, and then we will start talking about specific pathways and how the genes are transferred and what happen as a result of gene transfer, we will see in the subsequent classes. Thank you.